Alright, in today's video I wanted to spend a few minutes talking over the specific steps and details of the mitotic phase of the cell cycle. So let's go ahead and have a closer look. So this mitotic phase is sometimes also called just simply the M phase, or this is the step that we refer to as mitosis. So we'll use those words to describe this step interchangeably. This step of the cell cycle functions to divide up some chromosomes equally between two cells. So we're going to walk through the steps, and we're going to pay a lot of attention to chromosomes, what they're formed out of, how they're moved around, and how that divides these chromosomes equally between two cells during the process of cell division. So I will often summarize mitosis, or M phase, in a single sentence. And here's how I would summarize mitosis. I say that mitosis produces two genetically identical somatic cells. And one detail that I'll have you remember about somatic cells is that they are diploid. So diploid just means that they have two copies of each of their chromosomes. So as mammals, we are all diploid. We have two copies of every chromosome inside our cells. And we got one of those copies from our mom, and we got the other copy from our dad. So two copies, one from mom, one from dad. That makes most of our cells diploid. Most of our cells, I said, only our somatic cells are diploid. Somatic cells are all the cells in our body that are not either our eggs, if we are female, or our sperm, if we are male. So to summarize one more time, mitosis is a process that produces two cells. They're genetically identical, so they're both going to contain the exact same DNA. And we make somatic cells this way, all of our cells that aren't eggs or are not sperm. And those somatic cells are always diploid. They have two copies of each chromosome. So let's look more closely at where this mitotic phase falls in the greater cell cycle. So we'll talk more about the cell cycle overall, but here's an image of the cell cycle that breaks it down into this cycle. Cells progress through this over and over and over again. Every time a cell divides, it reinitiates this cycle and begins over. Today we're going to talk about M phase, this one little wedge right here. But M phase is just part of the cell cycle. The other part of the cell cycle, the rest of it, we refer to as interphase. So interphase is all the rest of the cell cycle that is not M phase. And interphase includes three parts. We refer to them as G1, S, and G2. We'll talk more about those steps and what's going on during them another time. Again, today we're going to pay attention to M phase, this one small part of the cell cycle. That part of the cell cycle, M phase, is broken into its own steps. So during the video, we're going to talk about prophase, metaphase, anaphase, telophase, and cytokinesis. All those steps are jammed into this M phase part. So together, M phase and interphase combine to make this complete cell cycle. And again, the cells progress through the cell cycle anytime they're going to divide. All right. So again, today we're going to talk about one part of the cell cycle, just mitosis, just M phase. And that's shown right here in these two cell drawings. And we're going to see that these chromosomes, which are these structures in the middle, are going to be manipulated, moved around. We're actually going to break them in half and separate them apart into these two cells that each have the exact same genetic content. They're genetically identical. But before we talk about the specific steps that occur during mitosis, I just want to remind you of one very important detail. And that's that DNA replication has to occur before mitosis. And to be more specific, that DNA replication step happens during S phase of interphase. So again, one part of interphase is S phase, and that's where we make a copy of our DNA. So that S in S phase actually stands for synthesis, and it's synthesis of DNA. So if we look at the pictures, our chromosomes start here as these bands, and when we go through S phase, represented by this arrow, we do DNA replication. And we make these chromosomes that are sort of X-shaped. So I want to talk a little bit more about that structure before we think about the steps of mitosis, so that we'll understand mitosis more clearly. So here's another drawing that represents those chromosomes before S phase, after S phase, and then after mitosis. So here again, I've got these bars to represent our chromosomes. This is in a somatic cell. Remember, somatic cells are diploid. They have 2 copies of each chromosome. So I've got my 2 different copies of this chromosome represented here by the 2 different color bars. Again, we would have gotten one of these copies of the chromosome from our dad, and we would get the other copy of the chromosome from our mom. So we refer to these 2 copies of the chromosome as homologous chromosomes. So we're going to use that term over the next couple parts of our course. Homologous chromosomes, the 2 copies of that same chromosome. Now, right now these chromosomes exist inside one cell, so within this circle. And then we do DNA replication, again, during S phase. And when we do that, our chromosomes double in size, and they're made of these 2 mirror images of one another. So these 2 structures are stuck to one another. And we call those structures sister chromatids. So after S phase, each chromosome is made of a pair of sister chromatids, these 2 structures stuck to one another. So this homologous chromosome has 2 sister chromatids, and so does this other homologous chromosome. It has 2 sister chromatids. And so as we go through the steps of mitosis, we're going to see that mitosis separates these sister chromatids into opposite cells. Each cell that we make during mitosis will get one of the sister chromatids. And at the end, we'll have 2 cells that each contain 1 sister chromatid, but 2 homologous chromosomes. And this cell is identical to the cell we started with in terms of the DNA that's present inside of it. All right, so that's all the information we need to know in order to talk about the steps of mitosis. So let's look at the stages of mitosis. We're going to break mitosis into 6 steps. We're going to think about what happens during each step and what cells look like when they're in each step of mitosis. So the steps are, first, prophase. Second, we'll talk about something called prometaphase. Third, we'll add metaphase. Fourth will be anaphase. Fifth is something called telophase. And the last step, number 6, is cytokinesis. So again, we're going to look at pictures of each step in M phase and think about what's going on during each step. So we'll go in order. We'll start with prophase. So here we have our cell. It's entering prophase, so it's leaving interphase, entering M phase, and the first step is prophase. So what happens during prophase? The first thing that we do is we take our chromosomes and we condense them. Basically, we pack our chromosomes into a smaller package. When you're moving, you do this kind of activity. You take all your stuff and you pack it into boxes because it'll be easier to move around if it's all condensed into a smaller package. And that's what our cell is doing. So we can see these chromosomes here in the nucleus. They are condensed down to small structures that we can observe. We'll look at some pictures later on. The other thing that happens is that a structure called the centrosome, there's 2 of them, are going to move towards opposite sides of the cell. So there's one centrosome here and another centrosome here. These structures are going to move towards opposite sides of the cells. So this one's moving over this way, This one's moving a little farther this way. These centrosomes serve an important purpose for the cell. They build this mitotic spindle, which is mostly built out of microtubules. So the little blue lines here are the microtubules, and they grow out of this centrosome. Again, we've got 2 of them right now. All right, so during prophase, we pack the chromosomes, we move these centrosomes apart, and we build microtubules. Next, we can move into prometaphase. During prometaphase, we're going to accomplish a couple more tasks. First, we're going to get rid of the nuclear envelope. So this little dashed line represents the old nuclear membrane, and we've removed it. Now the chromosomes are free to float around in the cytoplasm, and eventually we'll be able to move them to opposite sides of the cell. The next thing that happens during prometaphase is that the microtubules attach to the centromere of each chromosome. So again, my blue lines emanating from the centrosome grow outwards, and they connect to this part of the chromosome right in the middle called the centromere. So our microtubules grab onto the chromosomes. They grab onto the chromosomes at the centromere. So next we'll proceed into metaphase. During metaphase, the cell has a very specific appearance. And that appearance includes that all of these chromosomes have lined up right in the middle of the cell. And that's the main thing that happens during metaphase. All the chromosomes are pulled to the center of the cell. They're pulled there because every chromosome is attached to 2 microtubules, one from each side of the cell. And these 2 microtubules are pulling equally on that chromosome. I think of this step sort of like a tug of war. So during this tug of war, both microtubules are pulling. The chromosome gets pulled right to the middle of the cell. And that happens for all of the chromosomes. And that's metaphase. We also describe this lineup of chromosomes as the metaphase plate. So if you see metaphase plate, that just means the chromosomes lined up during metaphase. All right, the next step is anaphase. Here's what anaphase looks like. We can see our lineup of chromosomes is gone. Half the chromosome is over here on the left, half the chromosome is over here on the right. We've actually taken those sister chromatids and pulled them apart from one another. And the 2 sister chromatids from that chromosome are being pulled in opposite directions, one to the left, and one to the right. And that has happened for every one of our chromosomes. As anaphase ends, our chromosomes end up on opposite sides of the cells. And then we progress into telophase. In telophase, we reform the nucleus, the nuclear envelope, around our chromosomes. So we build one nuclear envelope on the left and one on the right to surround the chromatids that we move to opposite sides of the cell. In addition to remaking the nuclear envelope, we also unpack our chromosomes. We're done moving them around, so our stuff doesn't need to be in a box anymore. So we unpack it, and that way the cell can continue to use the DNA moving forward. Right? The next step, cytokinesis, is where we divide the cell into 2. So we've got our nucleus with the DNA in here. Again, it's unpacked now. You can't see the chromosome structures anymore. We've got a nucleus on either side of the cell. And during cytokinesis, we split the cytoplasm in half by pinching the cell apart right down the middle here. And that's going to create 2 cells that are genetically identical because they both contain one sister chromatid from each homologous chromosome. They each contain identical genetic information. So those are all the steps of M phase. And we'll continue to think about that moving forward. As always, I hope you'll ask if you have any questions, and I will talk to you soon.